Hello and welcome back to the Biochemistry for Health Sciences channel. In the last video, we talked about the structure as well as properties of two oxygen-binding proteins, hemoglobin as well as myoglobin. In this video, we'll talk about the physiological role of these two proteins, hemoglobin as well as myoglobin. In order to understand the functions of these proteins, we will consider two organs, the lungs as well as tissues, and an example of tissue that we'll use in this video would be muscles. Let's say the skeletal muscles. As we discussed before, the protein myoglobin is found in muscles, skeletal muscles as well as cardiac muscles, while the protein hemoglobin is found in red blood cells, which means it is found everywhere in the body, since red blood cells travel through blood vessels throughout the body. Let us first look at what happens to these proteins in the muscle tissue. The concentration of oxygen in tissues such as muscles is around 30 to 40 millimeters mercury. The pH is slightly acidic. 7.4 is the normal pH of blood. In tissues such as muscles, the pH is slightly lower at 7.2. So let's look at the key steps that happen in the muscles. So the reason why the concentration of oxygen is low in muscles and in tissues in general is because the oxygen is used up to generate ATP or energy, something we'll talk about in metabolism in future videos. And one of the products of energy generation is the gas carbon dioxide. As we mentioned in previous videos, energy generation happens all the time. Tissues do not store ATP, so the tissues such as muscles have to continuously generate energy, which means they are continuously generating carbon dioxide. Carbon dioxide is a hydrophobic chemical. It readily crosses the plasma membrane of the muscle cell. So it basically diffuses out of the muscle cell. The carbon dioxide crosses the interstitial space and then crosses the endothelial cell wall and finally reaches the bloodstream, the plasma. Once in the plasma, which is the aqueous or the liquid part of blood, about 10% of the carbon dioxide that is produced by the muscle cell actually dissolves in the water part of plasma. So carbon dioxide plus water will give you carbonic acid, H2CO3. The remaining 90% crosses the red blood cell membrane and enters the cytoplasm of the red blood cell. Once in the red blood cell, the carbon dioxide undergoes two major reactions. About 10% of this carbon dioxide will react with the amines of proteins such as hemoglobin in the red blood cell, and when carbon dioxide reacts with the amine groups of hemoglobin, protons are released and we get this product that we call carbamino hemoglobin. It's basically a carbamate chemical. The second major reaction that the carbon dioxide can go through in the red blood cell cytoplasm is this reaction that we'll call the carbonic anhydrase catalyzed reaction, or CAB reaction. So most of the carbon dioxide, about 80% or so, will react with water to give you carbonic acid, and this then breaks down to give you protons and the chemical bicarbonate. So this reaction is catalyzed by the enzyme called carbonic anhydrase. So since carbon dioxide produces protons, we say carbon dioxide is a volatile acid. And these protons is one reason why tissues have a pH that is slightly less than 7.4, so in this case around 7.2. The protons that are released from the carbonate reaction as well as the CAB reaction, these protons will bind hemoglobin. The carboxyl and the amine groups that are unprotonated will bind protons, and that will change the weak forces in the hemoglobin molecule, thereby changing the shape of the hemoglobin molecule from the R state into the T state. Because of the change in shape, the curve, the oxygen dissociation curve, will shift to the right. So the oxygen dissociation curve shifts to the right, which means the affinity, hemoglobin's affinity for oxygen decreases, as we mentioned in the previous video. Whenever we see the curve shift to the right, that means the affinity of the protein for the ligand decreases. So oxygen is no more bound tightly to the hemoglobin. The oxygen is released, and this oxygen that is released from the red blood cell will diffuse towards the muscle tissue where the concentration of oxygen is very low. So basically, binding of protons to the oxyhemoglobin HBO, which is in the R state, will convert HBO into HHB, which is deoxyhemoglobin. This is in a T state, and this will result in the release of oxygen. The removal of the protons, so as these protons here are removed by binding to oxyhemoglobin, the concentration of protons go down. So these reactions will go in the forward direction. So basically, more carbon dioxide in the red blood cell will be converted into the products that will decrease the concentration of carbon dioxide in the red blood cell, and that will enhance the diffusion of carbon dioxide from the muscle into the red blood cell. So removal of protons will increase the diffusion of carbon dioxide from the muscles into the bloodstream into the red blood cells. Note from the oxygen dissociation curve that at a PO2 of 40 millimeters mercury, so 40 millimeters mercury right here, if we extrapolate the curve to pH 7.2, the oxygen saturation is about 70 to 80%, which means in the muscles, the hemoglobin, only about 70 to 80% of the hemoglobin has oxygen bound to it. Due to the change in pH from 7.4 to 7.2 in muscles, we have about 10% more oxygen release under this lower pH compared to pH 7.4. So to summarize, the binding of carbon dioxide to hemoglobin, to oxyhemoglobin, basically increases the release of oxygen from oxyhemoglobin, and that oxygen will then travel to the muscles for energy generation. So very nice system. Muscles release carbon dioxide while the red blood cells release oxygen. So this effect, where the increased carbon dioxide in peripheral tissues causes a right shift of the curve, thereby increasing oxygen delivery to tissues, this effect is also called the Bohr effect. Besides carbon dioxide, there is another chemical that helps release oxygen from oxyhemoglobin, thereby delivering the oxygen to tissues, and this chemical is called 2,3-bisphosphoglycerate. Bisphosphoglycerate. When the PO2 is low, as what we find in tissues such as muscles, red blood cells use an enzyme called bisphosphoglycerate mutase, and this enzyme basically converts 1,3-BPG into 2,3-BPG. So in tissues where PO2 is low, the concentration of 2,3-BPG increases in red blood cells. Now 2,3-BPG will bind the central cavity of hemoglobin right here between the 4 subunits. And when 2,3-BPG binds hemoglobin, it stabilizes the deoxy form of hemoglobin, the T state of hemoglobin. And this once again causes the curve to shift to the right. That means the affinity of hemoglobin for oxygen goes down, oxygen is released from hemoglobin, and that oxygen will then get into the muscle tissue. It's therefore not surprising that if there is a deficiency of the enzyme bisphosphoglycerate mutase, or any of the other enzymes in glycolysis, which we'll talk about in the future videos, any glycolytic enzymes that lead to the formation of 1,3-BPG. If any of these enzymes are deficient, then the concentration of 2,3-BPG in the red blood cells will be low, which means the hemoglobin, the ability of hemoglobin to release oxygen will go down. So just to summarize, there are two very important reasons why the oxygen from oxyhemoglobin is removed when the red blood cells are in tissues such as muscles. One is the binding of carbon dioxide to hemoglobin, and the second is the binding of 2,3-BPG to hemoglobin. Both of these binding causes the oxygen to dissociate from hemoglobin. That oxygen will then be sent to the muscles for their use. Oxygen is also a hydrophobic gas. It will readily diffuse out of the red blood cell, get into the interstitial space, and then easily cross the membrane of the plasma membrane of the tissue cells, such as the muscle cell. Once in the muscle cell, the oxygen could combine with myoglobin to form oxymyoglobin. And of course, some of the oxygen can be used directly by the cell for energy generation. So muscle cells actually do store some oxygen in the form of oxymyoglobin. Another important process that occurs in the red blood cells when they are at the tissues is called the chloride shift. In order to prevent an increase in pH within the red blood cell, as the protons are removed by combining with hemoglobin, what happens in the chloride shift is bicarbonate that is formed is transported out of the red blood cell into the plasma. In order to maintain charge neutrality, since a negative charge is removed from the cell, another negative charge is brought into the cell, and this is your chloride. Your chloride enters in exchange for bicarbonate that is getting out of the red blood cell. So basically, the bicarbonate in the red blood cell is sent out to the plasma, while the chloride from the plasma enters the red blood cell. This transport is carried out by a transport protein that is called the anion exchange protein, AE1. So this AE1 kicks out the bicarbonate and brings in the chloride ions. This is called the chloride shift. So just to summarize, at the tissues such as muscles, carbon dioxide that is coming from the tissue together with BPG that is formed in the red blood cells, both these chemicals will help release oxygen from oxyhemoglobin, and that oxygen will then be transported to the tissue. So carbon dioxide will be taken up by the red blood cell, while oxygen will be given by the red blood cell to the muscle tissue. So at this stage in the muscles or other tissues, the red blood cell will have quite a bit of deoxyhemoglobin. Compared to tissues such as muscles, where the PO2 is only 40, in the lungs, the PO2 is much higher, around 100 millimeters mercury, and the pH is 7.4 compared to 7.2 or so in tissues such as the lungs. So let's see what happens to the hemoglobin when the red blood cells reach the lungs. So during inhalation, we take in oxygen, and that oxygen will enter the nasal oral passage, the trachea, the bronchioles, and eventually reach the alveoli in the lungs. The oxygen will then cross a thin membrane, and this membrane will consist of a layer of surfactant, cytoplasmic extensions of specialized alveolar cells known as type 1 pneumocytes, the basal lamina that is shared between type 1 pneumocyte as well as the endothelial cell, and the endothelial cell lining the blood capillary. In the plasma, very little oxygen, hardly any oxygen will dissolve in the water part of the plasma because oxygen is much less water soluble compared to carbon dioxide. So most of the oxygen will enter the red blood cell. So in the red blood cell, the oxygen will bind to the deoxyhemoglobin, and this will form oxyhemoglobin. Remember, this is in the R state of the hemoglobin. This is your T state, and protons will be released. In the previous video, we mentioned what happens when oxygen binds deoxyhemoglobin. Oxygen will enter the active site of the hemoglobin. The electron density around the iron atom will increase due to the presence of oxygen. The electrons in the d orbitals of the iron will be rearranged. The iron atom becomes smaller by about 19%. It moves into the plane of the heme, since this iron is connected to heme and heme is connected to the helix F8. The whole helix moves, and that causes a change in the shape of the hemoglobin from the T state to the R state. So the change in one subunit from the T state to the R state will cause weak forces such as salt bridges to break between that subunit and the other subunits, and now the other subunits will start changing from T state to the R state even without oxygen bound. This makes it easier for the other oxygen molecules to bind the hemoglobin because the rest of the subunits are now in the R state. This is called the cooperative effect. Another thing that happens is the cavity, the central cavity, becomes smaller when oxygen binds hemoglobin. And because now the cavity is too small, 2,3-BPG is removed from the hemoglobin. And due to the change of the hemoglobin, the entire hemoglobin molecule, the microenvironment changes, and the pKs of several groups, several amino acid R groups, will decrease, resulting in stronger acids, which means they will release protons. So protons are released when oxygen binds deoxyhemoglobin. These protons that are released from the hemoglobin will also promote the dissociation of carbaminohemoglobin. So these protons the increase in these protons will cause this carbaminohemoglobin to dissociate, releasing carbon dioxide from the hemoglobin. Bicarbonate is brought into the red blood cell by the AE1 transport protein, and this bicarbonate will then react with the protons that are released when oxygen binds hemoglobin, and this will cause the formation of carbonic acid, which will then break down into carbon dioxide and water. This reaction is catalyzed once again by carbonic anhydrous. So to summarize, in the lungs, when oxygen binds to hemoglobin, protons are released. Protons will help the dissociation of carbon dioxide from carbamino and hemoglobin. So we get carbon dioxide produced here. In addition, the protons will react with bicarbonate, producing carbonic acid, which will then dissociate and give you more carbon dioxide. So we get CO2 produced in the red blood cell. The dissolved carbon dioxide in the plasma, which exists as carbonic acid, will also break down into carbon dioxide. So all of this carbon dioxide produced by the red blood cell or the breakdown of carbonic acid will then be removed by the lungs into the atmosphere in exchange for oxygen. So to summarize the whole process, in tissues such as muscles, the carbon dioxide that is produced is taken up by the red blood cells in the bloodstream, while oxygen in the red blood cells is given to the muscle cells. In the lungs, where the CO2 is very high, the oxygen in the atmosphere is taken up by the red blood cells, while the carbon dioxide that they are carrying will be given to the atmosphere via exhalation. A few words about adult hemoglobin, which is HbA, myoglobin, as well as fetal hemoglobin, which is HbF. So here is the oxygen dissociation curve of myoglobin. This middle one is for fetal hemoglobin, and this one on the right is for adult hemoglobin. You can see that when the PO2 is very high at 100 in the lungs, adult hemoglobin is about 95 to 99% saturated. So this is about 95 to 99. Let's say 95. And when the red blood cells reach the tissues, where the PO2 is only 40 millimeters mercury, if you look at this point here, the YO2 or the saturation is only about 70%. So that's a difference of about 25%. So hemoglobin binds oxygen very well in the lungs and then releases it very well in the tissues. That 25% is released into the tissues. Now, what if myoglobin took over the job of hemoglobin in red blood cells? What would happen? Well, let's see. Myoglobin, that's this curve here, is about, let's say, 95-99% saturated at PO2 of 100, but which is the same as hemoglobin. But when you reach PO2 of 40, if you extrapolate 40 to the myoglobin curve, you see that this is still around 9900. So basically, myoglobin binds very well, but it doesn't release. Okay, so myoglobin does not release oxygen. It binds oxygen, but it does not release oxygen. And if it cannot release oxygen, then the tissues are not going to get the oxygen. So that's why myoglobin is not found in red blood cells. It's only found in the muscles, while hemoglobin is in red blood cells because in the lungs it can bind very well. In the tissues, it can release oxygen very well. So clearly the quaternary structure of hemoglobin is important for its function, not only for binding oxygen, but for releasing oxygen. That quaternary structure is not found in myoglobin. Myoglobin is only good in binding oxygen, but it cannot release oxygen. So this comparison of myoglobin and hemoglobin gives us a good idea of why some proteins have quaternary structures. A few words about the scenario in a pregnant woman. Fetal hemoglobin, the fetus hemoglobin called HbF, that's also tetrameric, has 4 subunits, alpha 2, gamma 2. Hemoglobin adult, the mother's hemoglobin, is alpha 2, beta 2. Now, interestingly, the P50 for hemoglobin F is around 19. The P50 for adult hemoglobin, the mother's hemoglobin, is 26. That means hemoglobin F binds oxygen better than hemoglobin A. So the fetus hemoglobin binds oxygen, has a higher affinity for oxygen than the mother's hemoglobin. The reason for this is because the fetal hemoglobin doesn't bind 2,3-BPG. It's poorly, weakly binds 2,3-BPG, and that's the reason why, remember, BPG shifts the curve to the right, but if fetal hemoglobin doesn't bind BPG, then the curve is shifted to the left. So since hemoglobin F has a higher affinity for oxygen than hemoglobin A, that means irrespective of whatever the PO2 is in the mother's bloodstream, hemoglobin F of the fetus can continuously steal the mother's oxygen for the fetus. So these dissociation curves of myoglobin and the various types of hemoglobin help us explain the physiological roles of these proteins. Before we end this video, a few words about the lab tests that are related to these last 2 videos that we talked about, today's video as well as the last video. There are several very important lab tests that are associated with hemoglobin and the transport of carbon dioxide and oxygen. One is the bicarbonate concentration. You can see it's about 22 to 26, so that's about 24 millimolar average. Then the PCO2, the partial pressure of carbon dioxide, 35 to 45, so that's roughly about 40 millimeters mercury. The PO2 concentration, 80 to 105. And another important lab test would be the pH. Blood pH is 7.35 to 7.45, so that's around 7.40. So we will use these values of bicarbonate, the PCO2, as well as the pH in future videos where we'll talk about the acid-base status of patients in health as well as in disease. That is all for now. We hope you have enjoyed this video as well as several others that we have in the past. Goodbye and have a wonderful day.